Welcome to another episode of Telikilaas biology period. I am Singh K. Anthony, biology teacher, Aminia School. We are going to learn today another subtopic under the unit inheritance. The subtopic is protein synthesis. Protein synthesis is taking place in all the cells in a daily basis based on the requirement of the body, and we need to learn the different steps involved in protein synthesis. Let us see the syllabus requirement of the topic. By the end of this lesson, you should know how to explain that the sequence of bases in a gene is the genetic code, and you should be able to explain that DNA controls cell function by controlling the production of proteins, and you should be also able to explain how a protein is made. So this is the requirement of the syllabus for this particular topic. Before going into the details of protein synthesis, we should have an understanding regarding the structure of DNA. The other day, the previous episode, we have defined the gene as the segment of DNA or the length of DNA that codes for a protein. So DNA has a major role to play in the process of protein synthesis. That is why we should have an understanding regarding the structure of DNA. And the structure of DNA was proposed by these two scientists, Watson and Crick, and their model is known as double helix model of DNA. So this is the double helix, and according to them, DNA has two strands which are spirally twisted together. That is why it is shown like this, two strands. But there is an exception. In 2009, a boy was born in England who is having an extra strand in the DNA. His name is Alfie Clamp, and he is the only human to have an extra strand in the DNA, particularly in the 7th chromosome. And they say it is an extra arm to the chromosome. Now coming to the structure. I have already told you that DNA is made up of two strands which are spirally twisted together, and according to their model, DNA is made up of smaller units called nucleotides. And if you examine the structure of each nucleotide is made up of a sugar molecule, and the sugar molecule is deoxyribose or deoxyribose, and that is why the name DNA, which starts with deoxyribose, then the nucleic acid. So the sugar molecule is deoxyribose, then a phosphate group and a nitrogen base. And the nitrogen base can be of 4 different types. It can be adenine, guanine, cytosine, and thymine. These are the 4 different nitrogen bases. When we see the picture, we will know about it. So again, the DNA strand is being shown. The nitrogen bases, adenine, thymine, guanine, cytosine, the names are being given. So each nucleotide is made up of a sugar molecule, a phosphate group, and a nitrogen base. And DNA is made up of many nucleotides. That is why DNA is referred as a polynucleotide. And this double helix model can be compared to a spiral ladder. and the upright of the ladder. Here is the upright of the ladder. The upright of the ladder is made up of the sugar molecule and the phosphate. And the rungs of the ladder is made up of the nitrogen bases. And the nitrogen base of one strand is pairing with the nitrogen base of the other strand. And they are not pairing as they like. They are pairing according to the rule of base pairing. According to the rule of base pairing, adenine will always pair with thymine and cytosine will pair with guanine. So adenine is always pairing with thymine. So when this strand is providing adenine, the other strand will provide thymine. When this strand is providing thymine, the other one will provide adenine. Likewise, when this is providing guanine, the other strand will provide cytosine. And when this strand is providing cytosine, the other one will provide guanine. And they are held together. These nitrogen bases are held together by means of hydrogen bonds. A double bond between adenine and thymine, a triple bond between guanine and cytosine. And the syllabus says not to go into these details, but an understanding of this is always necessary when we study protein synthesis, because the sequence of nitrogen bases are very important. So we have seen the structure of DNA. The other nucleic acid is the RNA, and RNA also having a role to play in protein synthesis. So we should know the difference between DNA and RNA also before going into protein synthesis. We have already seen that DNA is double stranded, but RNA is single stranded. Then we have studied that the sugar molecule in DNA is deoxyribose, and here the sugar molecule is ribose. That is why the name ribose nucleic acid. Then there is a difference in the nitrogen bases also. In case of DNA, adenine, guanine, cytosine and thymine. These are the 4 nitrogen bases. In RNA, thymine is not there. Instead of thymine, there is another nitrogen base known as uracil. DNA is only of one type, whereas RNA, there are 3 different types based on their functions. mRNA, which means messenger RNA, tRNA, transfer RNA, rRNA or ribosomal RNA. These are the 3 different types of RNA, and these RNAs are having their own role to play in the process of protein synthesis. Before going into the protein synthesis, we have already learned that the gene contains the genetic information. That is why the length of DNA or the DNA is considered as the genetic material. And scientists were trying to find out in what way this genetic information is being found. They knew that it has something to do with the nitrogen bases. And there are only 4 nitrogen bases, and if they are coding for amino acids, those 4 letters will not be enough. So here the genetic information in DNA is in the form of a code language, and it is referred as a genetic code. First they thought that it is only a single letter language. This adenine, guanine, cytosine and thymine, those 4 letters beginning at the beginning of this nitrogen bases. And there are 20 naturally occurring amino acids. And if these 4 letters are coding for amino acids, another 16 amino acids will be without any code. So they left the single letter language, went for the double letter. and when they are able to make combinations of these nitrogen bases to form doublets, they are able to get only 16 of them. Again, 4 nitrogen bases will be left without any code. So they came to the conclusion that the genetic code is in the form of a triplet codon. Triplet means, tri means 3, that means 3 letters there. And so the triplet codon is a sequence of 3 nitrogen bases in a strand of DNA, which is then copied down to form a messenger RNA. When we study the steps in protein synthesis, we will talk about it again. So just remember that the genetic code is in the form of a triplet codon. And what is this triplet codon? It is a sequence of 3 nitrogen bases in the strand of DNA, and that is being copied down into the messenger RNA. So genetic information is being copied down. So always remember that triplet codon forms the genetic code. And what does this triplet codon codes for? The triplet codon codes for an amino acid. Each triplet is coding for an amino acid during protein synthesis. For example, insulin. Insulin is a hormone, but chemically it is a protein. And insulin is made up of 51 amino acids arranged in a particular order. And that means in the DNA or in the messenger RNA, there are 51 triplets. I told you each triplet is coding for an amino acid during protein synthesis. If there are 51 triplets, that means there will be 51 amino acids to which these triplets coding for. That means how many nitrogen bases? Multiply by 3, 153 nitrogen bases. So just remember that each triplet codes for an amino acid, and based on the triplets during protein synthesis, amino acids are put together in the correct order and in correct number to make a specific protein. So always remember that it is based on the triplets which are found on messenger RNA. The protein synthesis will take place in the cytoplasm, and ribosomes are the site of protein synthesis. This is a chart showing the triplet codons, and I told you there are 20 naturally occurring amino acids. Corresponding to that, there will be triplets, and triplets are universal. That means when a triplet is coding for a particular amino acid in an organism, the same triplet will code for all the, triplet will code for the same amino acid in all the living organisms. So triplet is universal, and triplet is specific. When it is coding for a particular amino acid, it will code for only that amino acid. Here UUA is coding for phenylalanine, which is an amino acid. That means it will code only for phenylalanine. So triplet codons are specific, and based on the triplet codon, the sequence of triplet codons, the sequence of amino acids are also in the protein during protein synthesis. And how to stop the synthesis of protein? I told you insulin is made up of 51 triplets, 51 amino acids, and there are 51 triplets coding for those amino acids. And how to stop the protein synthesis? So in order to stop the protein synthesis, after the 51st triplet, there will be a stop codon. certain codons are stop codons or terminator codons. There are 3 of them. UAA is a stop codon, UAG is a stop codon, another one is UGA. So these are stop codons. That means after the 51st triplet, there will be any one of these triplet that will terminate the synthesis of protein. Understood? So always remember that the triplets, each triplet is coding for an amino acid during protein synthesis, and during protein synthesis, based on the order of the triplet and based on the number of triplets, amino acids are gathered from the pool of amino acids in cytoplasm to make a protein molecule. And coming to the second objective that is given here, explain that DNA controls cell function by controlling the production of protein. DNA controls the cell function, and always remember that all body cells in an organism contain the same genes. The previous episode we have studied about genes. Genes are found along the chromosomes, and 46 chromosomes will be there in all the cells. And always remember that all body cells in an organism contain the same gene. That doesn't mean that all these genes are going to express out in all the cells, but a gene will be expressed only when a particular cell needs to make a specific protein. And DNA controls cell function by controlling the production of proteins, and the proteins are being synthesized in a daily basis based on the requirement of the body, and these proteins can be enzymes. You have studied about enzymes in grade 9. Enzymes are biological catalysts. Then you have studied about antibodies, which are helping in providing immunity to the body. And also you have studied about neurotransmitters. Neurotransmitters are helping in the transmission of impulses across the synapse. You have studied what is a synapse, the gap between two neurons. Along the nerve fiber, the transmission of impulses is electrical. Across the synapse, it is chemical transmission with the help of neurotransmitters. Neurotransmitters will diffuse across the gap, and they reach the other side of the synapse, where the receptors are there containing receptor proteins. So the proteins which are being synthesized can be of various kinds. The syllabus says about these 3, namely enzymes, antibodies, and receptor proteins necessary for the neurotransmitters. And how the protein synthesis takes place. Protein synthesis takes place in 2 major steps, namely transcription and translation. Not necessary to learn those terms for your examination point of view. Syllabus says not to go into the details of all these steps, but you are supposed to know that transcription and translation takes place. What do you understand by transcription? When you were in lower classes, you might have written transcription in your language classes, where you have copied down something into certain transcription notebooks. Likewise, copying of the genetic information takes place. We have already seen that gene is the length of DNA, and that length of DNA contains the triplet, which is the genetic code, which is corresponding to the genetic information. And that genetic information will be copied down into RNA molecule. The RNA molecule that is being formed is known as the messenger RNA. So this happens within the nucleus of the cell. So formation of mRNA or messenger RNA takes place within the nucleus of the cell. From there, the messenger RNA moves out into the cytoplasm. And this copying down process is known as the transcription. And the messenger RNA that leaves the nucleus reaches the cytoplasm and get attached to the ribosomes. I have already told you that ribosomes are the site of protein synthesis. So what happens? The messenger RNA get attached to this is the ribosome, this is the messenger RNA molecule, and based on the triplet here, these are all transfer RNAs. They are able to recognize the specific amino acids. I told you these triplets are specific. They code for a particular amino acid, and based on that, the transfer RNA, they are able to recognize them, and they are brought into this site, and the assemblage of the amino acids takes place to form a protein molecule, which we call it as the translation. So the copied down genetic information is being translated as a protein molecule. So from DNA to mRNA and then to protein. So just DNA to mRNA, that is transcription, mRNA to protein molecule, that is translation. So this is how protein synthesis takes place, and the syllabus is asking you to, this is the diagram I have taken from a past paper question. Here we could see the DNA molecule, and this is the mRNA, which is copying down the genetic information, which is coming out into the cytoplasm, where it is getting attached to the ribosomes. So this is a diagram which is given in one of the past paper question. And these are the steps the syllabus is asking you to write in your answer when they are asking for protein synthesis. The gene that is coding for protein remains in the nucleus. It will not come out. The gene will remain within the nucleus of the cell. Then what is happening? Copying down, transcription takes place. Genetic information is copied to mRNA and carried to the cytoplasm. Then the messenger RNA or mRNA passes through the ribosomes or get attached to the ribosomes, and based on the triplets, the transfer RNA carry amino acids to the ribosome. Then the ribosomes will assemble amino acids into protein. The specific order of amino acid is always determined by the triplets which are found on mRNA. So this is how protein synthesis takes place. And if you are able to remember these steps, then you will be able to get all the marks when they are asking for protein synthesis. I have also added 2 past paper questions which you are supposed to answer these days. The first one, it is being found in May-June 2017 theory paper, question number 2, A and B subdivisions. The second one, October-November 2016, question number 2, A and B, there A is missing. So just remember that these are the 2 questions I have chosen for you to answer before you could listen to the next episode. Thank you for joining us with this episode. I hope you will stay indoors and break the chain.